Well, thank you very much for the invitation. It's quite nice. I wish I could be there in person. I do have a soft spot for Arizona, and I visited Flagstaff many times when I was living there doing my PhD, and I thought it was a most beautiful place. So you guys are all lucky to be there. Thanks for having me, and I'll go ahead and share my screen and see if everybody can see it. Can you see that okay? Okay. So the work I'm going to be talking to you about is work I did for my dissertation, which is really about this issue of bridging ideas in ecological theory with ideas in evolutionary theory. And these are the idea of merging ecology and evolution is not new, but I put somewhat of a new spin on it by focusing on fluctuating environments. And so I did most of this work in close collaboration with my advisor, Dr. Peter Chesson, who's kind of thought a lot about variable environment work for a long time. So if you're interested, I have a bunch of information here. You can contact me at the end or ask me some questions, and I'm happy to elaborate on anything that I talk about in the talk. So just to begin, here's a smattering of some of the organisms that I'm going to be talking to you about, which are annual plants that live in arid systems. So some of you who are better botanists than I am might be able to identify some of these plants very quickly, but these were the ones that I would go out into the field and study and use as inspiration for a bunch of theoretical questions. If you look at that particular system, or if you look at most environments around the world, you might ask the question, why are there so many species? On one hand, we have some very basic ecological theory that's quite old, and that says, that can be summarized by the competitive exclusion principle, that essentially says the species that are best adapted to whatever the environment is should exclude all others. And this was done originally in the 30s in a set of experiments with microorganisms. And you get something that looks like this. You have one in there, and you introduce another species, and it'll rise in abundance and drive the other one extinct. One of the real towering figures in our field, Hutchinson, formalized this issue in what's called the paradox of the plankton. So he wrote, the problem that is presented by the phytoplankton is essentially, how is it possible for a number of species to coexist in a relatively isotropic or unstructured environment, all competing for the same sorts of minerals? So he was thinking that phytoplankton, of which there are hundreds of species that you can find in a lake, were essentially following this competitive exclusion principle. And the fact that there were so many flew in face of that, these experiments and the theory around that. So if we're going to try and explain species diversity, at some level, it's actually fairly simple to understand. We think about the standing species richness in an environment. It depends on species inputs and species losses. Species inputs are things like speciation, so we get new species when they arise via the process of speciation. And then, of course, we can think about, for any particular defined area, expansion of a species outside of its range to a new range will introduce that species to the environment. So it'll be new. So the rates at which these occur will give us species inputs. And then, of course, species losses are given by extinction. For species richness to be higher in some area than in another, we can think about, we can have kind of this conceptual model where the inputs are relatively higher than the outputs of species. So any kinds of processes that increase inputs and reduce losses should elevate species richness in an environment. And most of ecology has focused around what are called species coexistence mechanisms and explaining the effects of species inputs and outputs in a system. Okay, so the definition here that I'm going to use, there are lots of definitions, but I like this one. It's any process that allows ecologically similar species, so like these birds that are over here, to persist together under defined ecological conditions. And so some of the traditional examples that you would find if you opened up a community ecology textbook are things like resource partitioning. So these birds are using different parts of the tree to find food, and so they don't compete for food within the same parts of the tree. Others that are really common or that you might be familiar with are like competition colonization trade-offs. So how do coexistence mechanisms affect species diversity? Well, they affect species diversity through changes in rates of species losses and species inputs. So the first thing that they do is that they delay extinction. So here I've just got a model, a hypothetical example of three species, this green, blue, and purple one, that are interacting with one another in a variable environment. So their populations fluctuate quite a bit. And here I've got a coexistence mechanism. And you can see that the three of them are bounded fairly strongly away from zero population size. So they're bounded away from extinction. Extinctions happen for every species that's ever existed on Earth, but we at least understand coexistence mechanisms to delay that process in some case. Now, if we have the exact same model, but without a coexistence mechanism, you can see that the two, what I'm just going to call inferior competitors here, the purple species and the blue species, they may not go extinct in this model, but they're hovering very close to this extinction boundary, and we would expect extinction to happen at a much faster rate without a coexistence mechanism than with one. Coexistence mechanisms also facilitate establishment of species in new areas. So here I've got these exact same species, but I'm going to introduce them in order of the purple, then the blue, and the green. And with a coexistence mechanism, shortly following introduction, they can rise in abundance and fluctuate around abundances that make them kind of safely established in an environment. If we do the same thing without a coexistence mechanism, you can see that following introduction of some species into a community with another one already present leaves it at relatively low abundance for a very long time period, and it may be susceptible to extinction in those environments. Finally, we think of coexistence mechanisms in an analogous process happening in the evolutionary space, where interactions between phenotypes within a species with the analogous coexistence mechanism can maintain polymorphism and be a prerequisite to ecological speciation. And so, you know, while species coexistence mechanisms um, uh, defined narrowly don't play any real role in speciation, the processes that are important also play a role in maintaining diversity within populations as well. And then there are lots of other reasons we care about species diversity besides just explaining what's happening in nature. One of them is that if we understand species coexistence mechanisms, we can manage for biodiversity. And there's a whole field of research in community ecology that is concerned with understanding how ecosystem functions, like here in this example, drought resistance, are related to species richness. It also helps us understand things like range expansion. So here's just a review article that was published a few years ago in Tree that essentially argued that to understand biogeography of a bunch of different species, you need to use the concepts in community ecology. Because species that extend their range are not extending their ranges into empty spaces, they're extending their ranges into spaces with ecologically similar species already present. And then finally, invasive species control is an obvious area where we need to care about species coexistence mechanisms, because when an invasive species rises in abundance in a particular community and then has large impacts on native species and drives them extinct, that represents collapse of coexistence of the native species with the invasive. So here's one of the particular problems with studying species coexistence mechanisms. This right here is a figure from Barrow, Colorado Island, where there are about 300 species of trees that are effectively all doing the same thing in the eyes of the ecologists who walk around there. Now, to explain diversity in the system, we can invoke species coexistence mechanisms, but which one? There are an enormous number of hypothesized species coexistence mechanisms, and I've listed just a few of them up here. A common one in this particular system is the Jansen-Connell hypothesis. There's resource partitioning of trees, plant-soil feedbacks, the storage effect, intermediate disturbance hypothesis, and I can go on and on and on. The issue is that an effective test of any one of these mechanisms is actually very difficult to do in the field. The experiments to design are very challenging. The manipulations have to be generally quite extensive, and the time to measure responses, especially for these tropical trees, can be really long. So the standard procedure is instead to ask, well, which one seems most likely for our system so we can focus our efforts? And we do this usually with two criteria. The first one is to ask which mechanism is most ecologically plausible given the community we're interested in. And then the next one is to ask which has the greatest potential to maintain diversity. You don't want to invoke a mechanism that only allows 2 species to coexist if you're trying to explain 300 species that are found on, on 50 hectares of tropical forest land. So I want to add a third one, which is the mechanism stable to evolutionary change via natural selection? The argument here is that natural selection is a pervasive force in biology, and that any mechanism, any species that are living in communities, we expect them to evolve to some extent in response to natural selection. And so as they evolve in response to natural selection, if the mechanism maintaining diversity of the species erodes and goes away following their adaptive evolution, we don't expect that mechanism to be prevalent in natural systems. So an example of this is one, again, we can pull from most community ecology textbooks, which is character displacement. Here's an example from the Grant's work on Darwin's finches, where we've got 2 species, Geospiza magnirostrus and Geospiza fortis. And they differ mainly in they're very similar, they eat very similar food, they feed on seeds, and what they eat depends largely on their beak sizes. So what they found and was reported in the science paper was that when seeds were abundant, these 2 species both had effectively fairly large beaks. And following a change in the environment where large seeds became very limited and small seeds were more abundant, there was a documented adaptive change in beak size for smaller beaks so that they could essentially consume these smaller seeds. And in doing so, a coexistence mechanism emerged, which is resource partitioning. So here's how this would work theoretically if we had lots of data. We could imagine these 3 hypothetical species that I showed you before here, where I've started them at initially the same abundance, and there's little to no or a weak coexistence mechanism among the 3 species. And over time, as they interact with one another, there's evolutionary trait change that leads to them to stably coexist with one another. And so we've got a strong, robust coexistence mechanism maintaining the 3 species here. So we know theoretically that this works for partitioning of substitutable resources, so things like seeds. And it also works for habitat partitioning. And of course, we have evidence of this in quite a few animal systems now. We know this for the finches, and we also have lots of examples of habitat partitioning in anolis lizards. So what about the other direction, where we have an established coexistence mechanism, and as the species interact with one another and evolve in the presence of one another, the mechanism erodes. We expect then for coexistence to collapse. Now, we don't have a lot of good evidence for this. I'm not sure how many people have actually looked for it, but we at least know that in theory, partitioning of essential resources look this way. So resource partitioning in plants, if they need nitrogen and phosphorus to grow and they have to eat both of them, they can't adapt for one to use exclusively nitrogen, the other one to exclusively use phosphorus. That doesn't work for plants. And then there's another coexistence mechanism called relative nonlinearity, where something similar works, and I'm happy to talk about that if anybody's interested. So that brings me to the mechanism of interest that I have, which is the storage effect. So the way you can understand the storage effect is that it's a form of partitioning of the environment in time. So the environment varies from time to time, and some species do better in some circumstances than in others, specifically in some environments over others. And that over time, when their environments are favorable, they can rise in abundance and in general be bounded away from extinction. So these dashed lines here give the exact same dynamics for the same model if we assume the environment doesn't vary. And so everyone experiences the same conditions from time to time. Exclusion happens very quickly. But if the species respond to the environment differently, they can coexist with one another. Lots of work on this particular mechanism shows that there are two requirements for it, which I'm going to talk about more in detail below or in a moment. But they're called buffer population growth and covariance between a species' response to the environment and competition that it experiences. And the question I have is this mechanism evolutionarily stable? There are a few theoretical studies on this, very few, like I can count them on one hand, and the results are mixed. And the reason is that fluctuating environments present unique challenges to organisms. The first, as I view it, is that selection varies over time, often unpredictably. And so there are some ideas out in the literature that when selection varies over time, it can be less effective. And when it's unpredictable, it also might be less effective. So part of this stems from the fact that the benefits to a particular phenotype occur only temporarily. When the environment varies, I mean, some phenotypes can be favored in some times and then others in different times. And so there's not particularly strong directional selection for one phenotype over another. And the last one is that there's another body of kind of demographic theory that says that demographic variation due to environmental variation is disadvantageous. It lowers the long-term fitness of organisms. And all of these suggest that a mechanism that depends on environmental variation might not be robust to evolutionary change. Okay, so that essentially brings me to the big question of the talk is the storage effect robust to adaptive evolutionary change of competing species? And I'm going to do this all theoretically, but I'm going to frame it in terms of real organisms where we have good evidence that the storage effect is relatively important. So most of you, I assume, are up here in Flagstaff. I did my dissertation work right here in Tucson at the University of Arizona. And over here, this picture is of Portal, Arizona, which is in the floodplain of the Chiricahua Mountains in southeastern Arizona. So it's right on the border between Arizona and New Mexico. And if the winter monsoons are good, you can go out in the spring and you might see something that looks like this, where all the vegetation that's short has lived its life in the past 5 or 6 months. So these are organisms that spend the vast majority of their life as seeds in the soil. And if the rain is right, they'll germinate and they'll grow extremely fast. They'll accumulate resources and then use up all of their stored biomass to produce seeds in enormous quantities. So here's a close-up of some of the species that we can find at that particular site in Portal, Arizona. And I'm just going to say right here that there are lots of empirical and theoretical studies that implicate the storage effect as an important mechanism maintaining diversity of these species. And part of that is simply because the environment is so variable and the species respond so strongly to it. If I go back to this picture, you can go out in a different year where the rainfall patterns will be different. You'll see a completely different assemblage of species that are there. And, at least at this site and another site in Arizona, at Tumamoc Hill, which is in Tucson, there are up to 40 or 50 years of demographic data on the occurrence of different species, and we can see that it varies a lot over time. And I should say here, because I didn't, that we know that there are at least 50 species of these annual plants over the space of just a few hectares in Portal, Arizona, and there are more than 150 species documented on a similarly sized area in Tucson. So these are extremely diverse areas over a very small space. The other thing that makes them really interesting for studying with respect to the storage effect is that both of those components I alluded to before are determined by a single trait in annual plants, and that trait is germination. So here's kind of a caricature of the life cycle of these annual plants. These are winter annuals, so in the fall, just before the winter monsoon rains come, all the plants are found as seeds in the soil. And then there's some winter rain, and the plant, each individual seed may germinate or remain dormant. If they remain dormant as a seed in the soil, they have to survive the entire year to be counted and then waiting for the monsoon rains to come again. If they germinate, they spend their winter trying to wait for the rain so that they can grow up and they grow big and they compete with one another They flower in the spring, they reproduce, and then all the seeds are set and then return into the soil to be counted again in the fall. So let's think about seed germination in terms of these two components that I alluded to. The first one I said was buffered population growth. And effectively what this is it's some form of population structure that reduces the effect of unfavorable conditions. And so in annual plants, we know that this is given by between-year seed dormancy. And so that means that this particular loop of the life cycle, where some seeds remain dormant every year and survive to be counted the next year, that loop exists for most of the species. And if you take a bunch of the species that are found in Tucson, and you try and germinate them either in the lab or in the field, it's actually very, very difficult to germinate all the seeds. They have some intrinsic reason to not germinate. And we think that part of that is adaptation for maintenance of this particular loop of the life cycle, which I'll get into. And you may see these really cool pictures around here. These are scanning electron microscope pictures of seeds of some of the species that you can find in these environments. And what I'd like to illustrate with some of these images is that the seeds are not monolithic, right? They're extremely variable across species in their morphology. They're extremely varied in their physiology. And we think that all of that variation goes into giving these different germination responses between species. So I want to give an example of how this works in terms of buffering. So what I've done here is I've simulated an annual plant life cycle, and I've partitioned fitness of any particular species into two components. The reproductive component, so this is the seeds germinating and growing up and producing more seeds. And then there's another component, which is seed survival. So if you're thinking of a perennial organism, seed survival of dormant seeds is effectively adult survival, the analogous adult survival for perennial organisms. If you look at the fitness, the reproductive fitness component of all of the fitness of a seed, it varies extremely wildly with environmental conditions. This year, it may be where there's lots and lots and lots of rain, so they can produce lots of seeds and reproduce quite well. But in other years like this one, they may germinate and there may be no follow-up rains. And most of the individuals may die following germination, which is a huge fitness cost for a seed because that's its only reproductive opportunity. At least at the lineage level, seed survival is an important fitness component that maintains fitness at kind of a minimum level, even if the environment's particularly poor, which is shown by this gray seed survival fitness component being above the reproductive fitness component. And so seed survival here is conditional on some of the seeds being dormant, and that buffers fitness from being too low, even when environmental conditions are very poor. So that's essentially how it works. The second component is what we call density-dependent covariance between environment and competition. We shorten that, like most scientists do, with an acronym to COVC. This is a statistical association between how strongly organisms respond to the environment and what effect competition has on them over time. In effect, what this component does is it says species, when at low density, are less regulated by competition when the environment is good. And so they can actually do better than their competitors at high density. And in kind of community ecology terms, what that means is that competitive effects between species are weak when averaged over time. And in annual plants, this is related again to their germination fraction, and it's given by species-specific germination responses. So this is data from, again, some of those species that you can find at Portal. And they've been, these are growth chamber experiments. So we have cold, we give them water under cold conditions, moderate temperature conditions, and then warm temperature conditions, which reflect some of the temperatures that they might experience out in the field. And then you just measure how well do they germinate. The colors here represent different species, and the main thing to take away from this figure is that some species that germinate quite poorly at cold temperatures germinate very well at warm temperatures. And those that don't germinate well at warm temperatures germinate quite well at cool temperatures. And so as the environment varies from year to year, you'll get different sets of species that show up in time, and this will give you your density-dependent covariance between environment and competition when coupled with buffered population growth. All right, so the approach I used in my dissertation was to investigate selection on both of these components and to ask, when are they favored under realistic ecological conditions? And if they are, we can come to the kind of conclusion that the storage effect is robust to adaptive evolutionary change. Previous studies haven't taken this approach. They've looked at which are partitioned by these components. So the first section is going to be about natural selection acting on seed dormancy. Can you hear me okay? I got a notification that my connection's unstable.
It just cut out for a second, but I think it's okay now.
Okay, so it's really about this fitness buffering. And then section 2 is going to be about natural selection on these species-specific germination patterns. So it really asks, are these patterns we observe in the field, do they persist under adaptive evolutionary change, or do they emerge from adaptation? So the evolution of seed dormancy works a little something like this. The kind of line on seed dormancy, this is something that's been studied in the field and theoretically for, we're going on 60 years now, almost 60 years, 50 years, I suppose. The argument is that seed dormancy is adaptive in a fluctuating environment because fitness is variable. And it's a bet hedging strategy. It's a way for organisms to diversify their fitness portfolio. So just like how when you're investing in the stock market, there are risky stocks and there are safe stocks. And the best thing to do is to allocate some of your money to risky stocks and then some of them to less risky stocks. That means you can maintain your money in risky years, but you can also make good headway and make lots of money on your risky stocks if the stock market goes up, and they do particularly well. So we think that a broad class of species and organisms are diversifying their fitness portfolios as well. So the evidence for this looks kind of this way, where we've got standard deviation or the variation in reproduction on the x-axis. We see that those species that experience greater variation in reproductive fitness also germinate at much, much, much lower rates than expected. And so they're heavily dormant, presumably because their portfolio is made up of mostly risky stocks, right? It's very risky to be the species once it germinates. Okay, and if you throw this into kind of an adaptive dynamics model and you make some predictions based on the field data, it looks pretty good, which is nice. The only issue I have with these particular studies, I think they're quite good, it's a minor issue, is that all these analyses assume that reproduction varies with the environment, but germination does not. And at least to me, the main thing when you go out from year to year and you look is that you get different numbers of species, different numbers of annual plants that show up from year to year. At least in popular press, we hear about super blooms, which has nothing to do with reproductive fitness of annual plants. That has everything to do with the fact that they germinate a lot more in some years than they do in others. So the question I asked in this first section is, this between-year dormancy that's required for the storage effect, is that adaptive when germination varies, either alone or also with seed yield variation? So here's some just details. Because this is science, I need to put what I'm actually doing up here so you can understand it. We've got a model of seed fitness that looks a lot like that life cycle diagram I have before, where we have seeds can either not germinate and then they have to survive, or they can germinate and they reproduce. They reproduce, that's based on the environment, which here is given by V, that's plant size in a year. This can be very big or it could be 0, that means they all die. And that declines with competition based on the total biomass of all the plants that are in the environment. So this is just a single species model. Germination and plant size both vary, and that's why they're indexed by T. We assume that germination follows a beta distribution. This is standard for things between 0 and 1. And the dormancy that we define is the temporal average germination fraction. And that's the parameter we let evolve in the model. And as another detail, we let plant size be log normally distributed. Okay, and the approach we took was one that's called adaptive dynamics. The highlights here are that it assumes that this is a mutation-driven process. It's not based on standing genetic variation. The mutations are small in effect, and they're fairly rare. And that means that evolutionary dynamics happen very slow relative to ecological dynamics. And in actuality, only selection is accounted for in this model, so any other evolutionary process, which we know is important, isn't in the model. Okay, so those things are important, but I haven't investigated them. It's complicated enough to do this, and maybe we'll add those in the future. And we considered 4 cases in the paper. One is where the environment is constant, the other one where the environment affects only reproduction or plant size. I'm not going to talk about those because that's 50 years worth of study. We just wanted to make sure that the model worked, and it did. I'm going to focus instead on these, where the environment only affects germination from year to year, or it affects both germination and plant reproduction, plant size. So here, let's look at this case where only germination varies. So I'm going to show a number of these kinds of figures, and they look like this. We've got essentially the evolutionary strategy at the evolutionary endpoint. Up here, we have no dormancy, and as we move down, we have more and more dormant types. And along the x-axis here, we have variation in the environment that affects germination. So as we move over here, the environment's constant. As we move right, there's more and more variation that affects germination. For statistical reasons, there's a region of parameter space here which is not feasible. It's just a minor complication that doesn't really change any of our results, but I'm happy to talk about that if people have questions. What we see is that with germination variation alone, fewer and fewer seeds germinate for the evolutionary end strategy on average in any given year. And so that means that dormancy is adaptive for any variation in germination. This result holds if we have 2 things. The first one is that seedlings, after they germinate, they compete for resources. And the other condition is that seedling competition varies. So in our model, this happens naturally. We've assumed that they've competed for resources. If we remove that assumption, we don't get any of these results. And seedling competition varies because the number of seedlings that are out from year to year varies with the germination fraction. In a hypothetical example where germination can either be 1 or 0, it could be everybody or nobody. If you flip back and forth between those years, there's high seedling density and then low seedling density. And that creates the variation in reproductive fitness that selects for dormancy. So if we compare the kind of selective effect of germination variance relative to reproductive variance, variation in germination is a much more powerful selective force acting on dormancy than variation in plant reproductive success due to environmental conditions while they're growing. So here, I want to show you that the y-axis scales are the same, and so you can compare these lines directly, but the x-axes are hugely different. So the variation here is on the log scale, and here it's 10, and this is 0.15. So these are enormously different, and it doesn't take very much germination and variation in germination to select for quite a bit of dormancy. So the last thing I want to say, and this is kind of a cluttered figure, but it shows the exact same result as I showed before here in this purple line. And if we add variable plant size due to, say, differences in rainfall from year to year, that's what we get more and more variable conditions as we move down here from purple to yellow colors. And so these 2 kinds of forms of variation add together to select for more and more dormancy. All right. So the conclusions from this section are kind of an obvious one, but it's been missed in a bunch of theoretical studies. Probably not because anybody who's doing this doesn't recognize it, but probably just because it was hard to do. This ended up being a lot harder than I thought it was going to be. The environment affects multiple aspects of an organism's fitness. Most studies of variable environments assume the environment affects one thing, right? One fitness component, but multiple fitness components, and they can interact and have differential effects on fitness. Each, in this example, can create reproductive risk that selects for a buffered population growth, a bet hedging strategy, dormancy. All of those are the same thing in this particular example. So we can conclude that dormancy and buffered population growth are evolutionarily stable in a fluctuating environment. Okay. So here's the summary. We can check this off the list. We could get selection on seed dormancy, and it required competition and environmental variability. That sounds great if we're interested in the storage effect, which is all about how environmental variability affects competition. Okay. So the next section is section 2 about natural selection on these species-specific differences. So I just want to remind you again that in the field, we find species that are very good at germinating under cold conditions and species that are very good at germinating under warm conditions. So they respond to the environment differently. And my question for this second section is, are these differences adaptive? There's an alternative explanation to them being adaptive, which is that they might have been evolved in some different environment when the species were not in contact with one another. And after they assembled together in the community that we see them in today, it happens to work that the two of them allow them to coexist. But they may not be a product of adaptive evolution in competition at all, right? So that's a viable hypothesis. It's also possible that once they come into contact with one another in this new environment, there may be an alternative environmental optima that they're both converging toward, but we just haven't seen it yet. And so as they converge toward that optimum, their differences might be muddled, and then we might expect collapse of coexistence. Okay, so in this model, it looks a lot like the last one, except now we've got two competing species. And I've made a subtle difference here. We're no longer going to model germination as beta. It's going to be normal on the log odd scale. So if you're doing a logistic regression, this is what your computer is doing. It's assuming that your things that happen between 0 and 1, your probabilities, are normal on the log odd scale. And so if we look at just a hypothetical distribution of germination fractions over time, this is what that distribution might look like. And the reason why I do that is that there's a nice, clever way, an easy way of modeling temporal patterns of germination under that particular model. And we're again going to assume that plant size is log normally distributed. And so a distribution of plant size might look something like this if it's log normally distributed over time. So 1 year, plants essentially don't grow very large at all. And in very rare cases, they are enormous. And most of the time, they have some average value that's intermediate between those two. Okay, so the model again has seed survival of seeds that don't germinate. The seeds that do germinate grow and to some species specific effect. And then there's competition from both species that are in the model. Okay, again, for the analytical approach, we're going to assume that evolutionary dynamics follow the adaptive dynamics assumptions I talked about, rare mutations of small effect that limit evolutionary change. And the way we did this was that we compared evolutionary endpoints, evolutionary outcomes in sympatry and allopatry. This is really commonly done for models of character displacement. And for those of you who are not familiar, it works like this. An allopatric situation is where we consider adaptive evolution of each species alone in their environment. And then we compare that evolutionary outcome when the ranges of the two species overlap, and we look at the two species in contact with one another. Do their adaptive evolutionary trajectories change when they're in contact? And whatever the change in the evolutionary outcome is, we can attribute to interspecific competition between the species. All right, so back to the question, are species differences in germination strategy adaptive? The answer is yes, provided, okay, there are always some provisions here. It doesn't happen under every scenario, but these are the scenarios. A seed cannot germinate equally well in every environmental condition. So what this means is that the seeds face some trade-offs. They can't germinate really well under cold conditions and also under really warm conditions. There's some physiological mechanism that limits that. We don't know if that actually happens we don't know if that constraint exists in real seeds. It's common for them to show some constraints, but we don't know if that's an adaptive outcome or if it's some physiological constraint. So The other provision here is that competition has to be intense, and so that means that interactions between species have to lead to some measurable fitness effects. And this is what it looks like. I've reformulated the model so that we're just going to think about cold germinating species and warm germinating species and those that kind of do with something in the middle. If this is the optimal environment, we start with the two species together, a blue and a green one, and we let them run over evolutionary time. This is the number of mutations. They diverge so that one specializes on germinating in cold environments and one specializes on germinating in warm environments. And this is an example of character displacement in a temporally variable environment. So we can do some math by deriving a selection gradient, and we can show that there are two components of that selection gradient. This looks ugly, but it's two things that are quite simple. The first one is an organism's fit to the environment. And so that means that there's some selection for organisms to evolve to this particular value. Okay, we can move it around and they'll move towards that value. In opposition to that, so this is a plus sign and this is a minus sign, in opposition to that is character displacement, which causes the two species to move away from one another. All right? One thing I wanted to highlight here is that in front of this bracket, there's a multiplicative term here, which I've called gamma, which in the model is seed dormancy. And so for any of this selection to matter at all, they have to have some seed dormancy. And so the first result we got out of this, just from deriving the selection gradient, was that buffered population growth, which is a requirement for the storage effect, is required for there to be any selection for species differences, which we thought was kind of cool and neat. Now there's a complication here. If you know these, if you're a plant physiologist, you might ask, yeah, Nick, you're doing a lot of stuff about growth after they germinate, and we know a lot about that. Here are some interesting physiological facts we have about these plants. They respond to the growing conditions really differently. We know there's a trade-off between how fast they grow for a fixed number of resources. So as you move up this axis, they grow really, really fast. They accumulate biomass quickly. And they trade off that with drought tolerance. And so as we move from the left to the right over here, on the right, these have very high water use efficiency. And so they can assimilate biomass with very little water. And so we know that the species that are very drought resistant grow very slowly. And the ones that grow very quickly are not very drought resistant. And so if there's a year where there's lots of rain, these species will do very well. And if there's a year where there's little rain, these species will do very well. And so that'll give them different distributions of plant size over time. In one particular year, this one will have maybe low plant size, and this one will be larger. Okay, so the next question was, do these differences of the species have any selective influence on these differences that I just showed you are adaptive? And the answer is yes, and in two different contrasting ways. So in one case, they can inhibit it. So we can think about, again, sorry, I've got this backwards here. So do those differences inhibit character displacement that I showed you before? And the answer is no. And so the way to explain this is to show you another figure like I did before. This was a V shape before. So as we move through time, they diverge from one another. In this case, selection is much weaker on divergence. Competition is much weaker. And so the two species mostly converge on the exact same trait. And so we wouldn't expect them to coexist for very long in the same environment. There are no differences between the species here. Now, if we allow them to have different values on that space of the water use efficiency relative growth rate trade-off, which are depicted here by two different dash lines for the different species, they not only adapt towards those optima, they move even further away from the optima. So they're more divergent in their germination patterns than they are predicted by their plant growth strategies. So we take this to mean that that differences between species in their physiology can facilitate selection for divergence in their germination responses. Okay, so a way to summarize this is again to think, we're going to put here on the x-axis the differences between the two species in their growth strategy. So over here at 0, you can collect any two species like this Plantago and this Pecticaria. They're very close along the trade-off. And so over here along the x-axis, we're thinking about these two species. Over here at the max, we're thinking about this species over here as compared with this one. So they grow very differently. And then we're going to plot that against their evolved differences in their germination strategy. So whether or not they fall very closely along this trade-off of germinating well under cold and warm conditions. So I'm just going to show you here that as we move for the species being more different in their physiology under the model, this is the allopatric evolution expectation. So this is how they would evolve in their fit to their environment. They're not in contact with one another. And when we put them in contact with one another, we're going to ask, are they more different in sympatry, or are they less, or are they more similar? If they're more similar, that means that these differences here in growth strategy undermine character displacement and actually cause character convergence. And if the points fall up here, then that means that they facilitate character displacement. That these pre-existing differences between the species and their physiology favor and facilitate the evolution of differences in their germination patterns. And this is what the results kind of look like. No matter how you do the analysis, all the points are going to lie above this allopatric evolution line. And we get different shapes of these curves, which represent either weak or strong associations between the environments that control germination and vigor. So you could imagine that if there's a strong cue in the environment, that association would be strong. That plants would have some information about growing conditions when they're germinating. But if that cue is very weak, we have very strong divergent selection to begin with. Okay. So some conclusions from this section. That whole analysis back there, what I take away from it is that some combinations of species may assemble and stably coexist following selection, and others may not. And the ones that may not are the species that are very similar. So these down here on the trade-off, if they're very similar, we might expect them to converge on the same. The analysis suggests that in some cases they should converge on the same value. So down here, right, if they're very similar, we get no differences between the species that evolve in their germination strategy. But if the species have pre-existing differences between them, that can facilitate evolution of differences in other traits. Okay, so just a tantalizing observation from the portal set of species of annual plants is that we have, I think out of those 50 species I mentioned, we have 2 congeneric pairs. And one of the pairs is between a native and invasive species in the same genus. And so what we have are species with disparate evolutionary histories that are coming together. They have extremely different life forms. We haven't done the entire analysis on their physiology, but we suspect that most of the species have quite different physiologies. And that may facilitate differences in their germination that we see. Okay. So check both boxes here. The first component was favored by competition and environmental variability. The second component, these species-specific differences, are facilitated, but are made possible by dormancy, which is the first component. And again, competition and any pre-existing differences in other life stages of the organisms. And so we can check both of the boxes. And the way I see this is that we have feedbacks, selective feedbacks between requirements and selective agents of those requirements for the storage effect. So this takes us back to Hutchinson. His question was, how do we explain species diversity? I think that this and previous studies argue that the storage effect is a plausible mechanism that checks off a bunch of boxes. The first one is that in principle, okay, so there's an important caveat I'm going to have here, it can maintain diversity of any number of species. So in theory, right, I could construct a situation where we have 10,000 species that coexist. They may be fleetingly so, but it's possible. And that's much more robust than many other mechanisms. The requirements of the storage effect are from a bunch of other studies. We've shown that they're ecologically reasonable, and at least I hope I've argued that they're evolutionarily stable. So here's that important caveat. Other mechanisms are almost certainly important in any particular system. And so I'm not saying that the storage effect is the only thing. And also the storage effect is also certainly unimportant in some systems. It's not going to be the tell-all, but I think it might be an important one. And of course, there's more to the story. I alluded to this, but the evolutionary model is, to put it mildly, limited. So it assumes that mutation is the rate limiting step of evolution. So there's no standing genetic variation in the populations, and we know that's probably not true at all. The model has no genetic constraints. And so to feel comfortable with some of these conclusions, we'd want to subject it to some more realities of the evolutionary process. The other thing you might be saying is, yeah, Nick, that's nice. You talked about Tuamac Hill having more than 150 species and Portal having 50 species, and you showed us a model of ecological divergence, of evolutionary divergence of 2 species. And that's a fair criticism. It ends up being much, much more complicated to do this for many more species, but it's something I'm working on. So this is a first step. The other thing is that we've omitted a bunch of important ecological factors, especially for annual plants. So seed enemies are a huge thing out there. So we've got rodents. There's a whole other group here at UF that studies just the rodents that are inside of these plots out here. They're enormous plots, and they've done very, very good work to show that these things have enormous fitness effects on plants. And we haven't included that in the model yet, and they may change things in important ways. Mainly because these have a much different generation length than what we assume the annual plants have. And so the feedbacks, the selective feedbacks, may be decoupled in time. There are also fungal pathogens in the soil that have important fitness effects that we don't know a lot about. There's a whole other group of people that think that dispersal and spatial variation are extremely important in annual plants, and they probably are, and we haven't incorporated in any of that. And the reason that's important is that dispersal can also act as a buffering mechanism, as a bet hedging mechanism. And so that's nice, except that it undermines selection for bet hedging in terms of dormancy. That they don't add together, they don't complement one another. And we also know that climate change is an important force in these environments. These long-term experiments I mentioned have documented changes in the composition of communities. And so I'm hoping to extend some of this work to ask if the species are adapted to one another, does that limit their potential to adapt to a changing climate? And with that, I want to thank my collaborator and advisor, Peter Chesson. Lots of helpful feedback from my committee members and my funding sources. And I'll take any questions if you have them.
Thank you, Nick.